Treating genetic diseases at their root requires biologists to reach beyond simply reading the genome. They must actively control it, using molecular machines to target and rewrite the genetic code inside living cells. Most current methods rely on borrowing tools that already exist in nature, repurposing evolved systems like CRISPR or zinc fingers to perform new tasks. These natural tools are often clunky and family-specific, making them difficult to deliver effectively into human cells. Because they evolved for specific biological niches, they aren't easily adapted to target every unique sequence in the 3 billion-letter human genome. To bypass these biological constraints, scientists are working to design entirely custom, highly targeted proteins from scratch. This is known as de novo design. True mastery over the genome requires a transition from biological scavenging to precise, ground-up computational engineering. Designers face a massive physical roadblock. The structure of DNA is incredibly uniform across almost all sequences. This regularity stands in sharp contrast to the wild and diverse shapes that standard proteins form, which are filled with unique pockets and intricate geometry. Because the double helix looks so similar from point to point, the chemical differences between one sequence and another are vanishingly subtle. Designing a protein to grab just one specific sequence is like navigating a massive maze where every corridor, junction, and surface is visually identical. Due to this uniformity, previous lab efforts relied on massive, time-consuming physical trial and error, testing millions of variants just to find a single working match. To move past this physical bottleneck, scientists realized they needed a reliable, step-by-step computational method to guarantee a match before a single protein is synthesized. The Baker Lab addressed this challenge with a two-part computational assembly line, starting with Phase 1, the binder block. Researchers use RF diffusion 3 to generate a cloud of potential protein structures, refining this density through denoising steps until it wraps snugly around target DNA. Secondary tools then assign the amino acids, while AlphaFold 3 predicts if these generated puzzle pieces will actually fit together in a real-world complex. The researchers discovered that while these generated proteins bound to the target DNA, they were also promiscuous, meaning they would stick to almost any DNA sequence they encountered. Generating a custom shape that fits DNA is only a partial solution. The true engineering challenge is forcing that protein to ignore everything else. The team solved this promiscuity problem by adding a second phase, the specificity block. This is negative design. Instead of only checking for a good fit, they forced AlphaFold 3 to fold their custom proteins against the wrong DNA sequences. To filter the results, they used a mathematical metric called delta-min-PAE. The algorithm subtracts the confidence of the protein binding to the wrong DNA from the confidence of it binding to the right DNA. The result is a clear score for specificity. If a protein design shows a high affinity for the wrong DNA, it is discarded, leaving only the most selective designs for physical testing. By explicitly teaching the algorithm what not to bind, scientists can computationally guarantee a protein's precision before starting an experiment. To validate the pipeline, the scientists synthesized their top 96 digital designs to see how they performed in actual lab conditions. The results showed a nearly 100-fold improvement in the success rate of finding sequence-specific DNA binders compared to previous design efforts. These custom proteins achieved tight binding affinities that rival nature's best transcription factors, proving they are as effective as they are specific. This approach moves the field beyond the limited toolkit provided by evolution and replaces manual trial and error with computational certainty. By establishing a reliable pipeline for de novo binder design, this work creates a foundation for the next generation of highly specific, programmable genomic medicines.